Welcome back to Acing AP Biology. In this lesson, we will go over the basics of cell division in prokaryotes and eukaryotes. Recall the idea in cell theory that all cells come from pre-existing cells. Cells replicate through cell division. In unicellular organisms, duplicate offspring is created through cell division. In multicellular organisms, cell division allows for growth, development, and repair. In the process of cell division, DNA is replicated so each new daughter cell receives identical DNA. The genome is all of the genetic material in a cell of a particular species. Multicellular organisms have large genomes, while unicellular organisms have small genomes. Mitosis is the process of cell division in eukaryotic cells, producing genetically identical daughter cells from the original zygote. A multicellular eukaryotic organism begins with a single cell called a zygote, and through mitosis it grows into trillions of cells. The cells become specialized due to differential gene expression. Prokaryotes use binary fission to reproduce. Prokaryotes' genetic material is held in a single circular chromosome with double-stranded DNA and some protein. DNA is held in the nucleoid region. Binary fission is a form of asexual reproduction, where bacteria replicate their chromosomes and divide the two copies of chromosomes between the two daughter cells. It is a much simpler and much faster process than mitosis. The process of binary fission begins at the origin of replication, where the bacterial chromosome begins to replicate. Then the chromosome replicates, producing two copies of the origin of replication. The two copies of the origin of replication then move to opposite ends of the cell, using actin-like proteins. The bacterium becomes longer and elongates. The plasma membrane begins to pinch inward, and the cell begins to divide. Finally, binary fission is complete with two genetically identical daughter cells. Next, let's talk about chromosomes in eukaryotic cells. First of all, chromosomes in eukaryotic cells are linear. Chromatin is the uncondensed version of chromosomes composed of DNA and histone protein. When genetic material is in the form of chromatin, transcription can take place, starting the process of protein production. During mitosis, DNA is condensed into chromosomes, so transcription cannot occur during mitosis. Chromosomes are visible under the light microscope, but chromatin is not. Each chromosome is made up of double-stranded DNA molecules in segments called genes. Humans have 46 chromosomes in somatic cells, or non-sex cells. They have 23 chromosomes in gametes, which are sex cells like ovum and sperm. As you can see, a eukaryotic chromosome is linear. It is composed of chromosome arms and the centromere, which is in the center, and the centromere contains protein kinetochore. During DNA replication, also known as S phase, identical chromosomes are paired in sister chromatids. They are attached to each other using kinetochore protein in the centromere. During mitosis, sister chromatids split, and each new daughter cell receives identical chromosomes. Keep in mind, when counting chromosomes, count the number of centromeres. The number of chromatids will be double this. That's it for this video. In the next lesson, we will go over the details of the cell cycle. See you then. For more bio content, hit subscribe.